Yes, where we have stopped in the last class. Transcription over. तुम्हारी वो तुम्हारी बैग वो. Yes, shall we start? Yes. So in the last class, we were discussing about the transcription process. Yes. What exactly transcription is? Can you define? Last bench. Can you define? Yes. Not audible. Yes. Can you define what is transcription? You can come front now. Last three. Come. I saw a picture betting. Come front. Yes. So So already I have told you about the central dogma of molecular biology. Yes. So DNA, with the support of DNA, mRNA, yes. So here, when we study transcription process, no, we have studied about formation of from the DNA synthesis of RNA. Ne? Which RNA? It can be mRNA, it can be tRNA, it can be rRNA. No. So here, this is transcription. Yes. Now, from this mRNA, when we synthesize protein, when we produce protein, no, the process is called translation. No, in short, translation means protein synthesis. Protein synthesis. Yes. Now, once we know the central dogma of molecular biology, it is completing by two processes. One is transcription, one is translation. Already we know what is transcription. Whatever the information that is present in the parental strand of the DNA, that is copied by the mRNA, and RNA is synthesized. Yes. Now by taking that RNA which is synthesized, we need to produce a polypeptide chain or a protein molecule. That whole process of making a polypeptide chain or a protein molecule with the support of transcription is called as translation. Right? Yes. The question arises where this translation process will take place. Before going to the process, any idea? Where is transcription happening? Yes, inside the nucleus. Nay. Now where translation will happen? Yes, outside the nucleus. Huh? What is present outside the nucleus? Yes. So translation process will take place in the cytoplasm. It will take place in the cytoplasm. Yes. Now coming to the process of translation. If you recall transcription, to conduct the transcription process, we have transcriptional unit. Yes? Nay? What is the transcriptional unit made up of? In the last class we studied. Nay? A promoter, structural gene, and the terminator. Yes, together makes the transcriptional unit. Right? So similarly, to conduct the translation process, yes, you can listen. Don't write anything. Listen. Okay, if you understand, you can write by your own. Yes, don't write anything. Listen, because it is a lengthier process. If you understand each and every point, then only finally you will understand the whole process. If you miss something, everything will be a confusion. Is that clear? Yes. So, coming to the translation process. To undergo with the translation process, again we want a translational unit. Without the unit, it is not possible. Nay? So, this translation process will be carried by translational unit. So, this translation unit consists of, number one, start codon. Number two, amino acid transferring site. Number three, stop codon. And number four, UTR. Four things are together makes the translation unit. Is that clear? So where this translation unit is present, this is present on the, this is present on, from the transcription what we have made? We have made RNA, no? So now I will take mRNA. This is our mRNA. On this mRNA, we should have all these four. Okay, we should have all these four. So here assume AUG, which is the start codon. Yes. So we have discussed genetic code. No, it is not finished. Yes. So when we study genetic code, totally how many codons are present? Any idea? Yes, 64 codons are present. Yes, we'll go to that later. In that 64 codon, 61 will be coded and 3 are nonsense codon. Okay. So that 3 nonsense codons are stop codons, we will tell, that is UAA, UAG, UGA. Yes, 3. So anyone, if anyone is coming in the process will stop there itself. Okay. So here assume, for example, here UGA is present. Yes. So I have taken the structural gene. What is structural gene here? mRNA. mRNA with the coding sequence is the structural gene. Now, the start codon, I have taken start codon, I have taken stop codon, no, and amino acid transfer site means the amino acid should get converted to peptide. It should get converted to peptide. First peptide, then polypeptide, then finally protein molecule. Okay? So here, structural genes with in the mRNA, start codon, stop codon, I have made. Okay. Now we have studied two types of transcription process, if you recall. Transcription in prokaryotes, transcription in eukaryotes. Okay. Transcription in prokaryotes was very simple. Initiation, elongation, termination, finish. Whatever the information that was copied, finally mRNA was produced. Yes or no? Yes. Second, we studied the transcription process in eukaryotes. In eukaryotes, what has happened? Additional process was added. Why? What was the reason? Because in prokaryotes, we have polycystonic genes. Same. Here, monocystonic genes. Introns are also present, exons are also present. So what we did, three process we took: splicing, capping, tailing. Yes. So in the 5' I will draw the capping also. I have drawn one cap. Yes. And coming to the tailing part, polyadenylate residues were added. So you can add the polyadenylate residue. Yes. Done. So this is the translational unit. It is not there in your books. It is not there in Jivit also. It is not in any book also. Okay. This makes the translational unit. So here promoter, the work of the promoter is done by start codon AUG. Terminator, stop codon. Is that clear? Separately promoter and terminator is not present. It is not present. Why? Because in eukaryotes, RNA polymerase will do its own work. It will do its own work. In the prokaryotes, to the RNA polymerase, factors are required. Sigma factor, RHO factor is required. But here, there is no necessity. So therefore, promoter and terminator is not present. Instead, start codon and stop codon is present. Done? Yes. This is the first translational unit required for translation process. Yes. Apart from that, what is needed? Any idea? Yes. Any other thing needed for the process? No? Yes. Second thing, no? Where the process has to take place? Yes. It is taking place in the cytoplasm. No? But where proteins get synthesized? Yes. No. There is, if you take any cell, we are talking about eukaryote, take our cell itself. No? Our body, our cell. So in our cell, in the cytoplasm, there is one small molecule made up of a protein, a small molecule made up of protein plus RNA. What is this called? Yes. No. It is called ribosome. What is ribosome called? Protein factory. Nay, in our body, ribosome is called as nay. In each and every cell where protein synthesis will take place, where ribosome is present. Nay, you have studied about endoplasmic reticulum, smooth endoplasmic reticulum, rough endoplasmic reticulum. The rough endoplasmic reticulum binded with the ribosomes. What they are doing? They are supporting protein synthesis. Without ribosome, protein is not synthesized. So therefore, smooth endoplasmic reticulum, what they do? They will undergo with the lipid synthesis. Is that clear? So second thing what is needed is the ribosome. Is that clear? Now, what is ribosome made up of, sir? It is made up of protein. It is made up of rRNA, ribosomal RNA. Is that clear? So here the prokaryotes also have ribosome, eukaryotes also have ribosome. Is that clear? But what is the difference between prokaryotic ribosome and eukaryotic ribosome? So usually the ribosome is classified into two units. One is larger subunit. Yes. And one is smaller subunit. Larger subunit and smaller subunit. So the prokaryotic ribosome consists of 50S and 30S subunit. And if you take eukaryotic, this is prokaryotic. If you take eukaryotic ribosome, 60S plus 40S ribosome. Is that clear? Overall, the prokaryotic ribosome is 70S subunit. And this is 80S subunit. Calculation is correct? Yes? Calculation is correct? Yes? There is no calculation required here. No. What is it? 50S, 30S? Listen here, don't calculate anything. Yes, tell. What is 50 and 30? Yes. When we take a prokaryotic ribosome, ठीक है, put it in the centrifugation machine, एक centrifugation machine में डालो, ठीक है, mix करो उसे, ठीक है. When the ribosome come and settle, when the ribosome after sedimentation, when the ribosome come and settles, what is the speed? ठीक है, that speed is that S unit. That S stands for a scientist by name Swedberg unit. Swedberg is the scientist who has calculated the sediment rate, at what level the sedimentation will take place, at what speed it will go and settle. So here it was able to see in the prokaryotes, the larger subunit will come and settle at 50s, okay, and smaller subunit will come and settle at, okay. Then why this 70, sir? This is average, approximate. Exact नहीं बोल सकते ना, नहीं तो approximate it is 70s. Therefore it will tell us the prokaryotic ribosome is 70, it is having 70s subunit, and 80s here high speed में settle हुए तो 60s, smaller 40s, ठीक है, slow rate में, together approximate is 80s. Did you understand? So in both same ribosomes are found. The ribosomes of the prokaryotic and eukaryotic cell has same sublevel, smaller, higher, everything is similar. Is that clear? So second thing what is required for the process is ribosome. Done? Ribosome with the larger subunit and smaller subunit. Larger subunit is 60s, smaller subunit is 40s. Okay. What is ribosome made up of? It is made up of RNA, rRNA and protein. Okay. Approximately how many proteins are there? In few books it is given 80 proteins are present. 80 molecules of proteins are already present. Is that clear? Second. Done? Now, before going to the next part, sir, why we are discussing, if you know all the requirement, what is involved, easily process will over. Okay. So first thing, translational unit we have studied. Second, ribosome. In the ribosome, when we study ribosome, what is the main thing we have to study? Inside the ribosome, no? Inside the ribosome, we are able to see three sites. We are able to see three sites. Very careful. This is the main thing. Okay. In the ribosome, we have three sites. Okay. What are the three sites, sir? This is called as A site. This is called as A site. This is called as P site. This is called as P site. And this is called as E site. Three sites are present. What they will do, I will show you in the explanation part. Done? So, second one we have done. Okay? Number one, translational unit is required. In that, we have mRNA. We have taken mRNA, then start codon, stop codon, and one thing I left, UTR. Nobody asked. नहीं सर क्या भी समझ में नहीं आ रहा, क्या पूछना? नहीं ध्यान था ना? Yes, I told you what start codon will do, what stop codon will do, and what will happen to amino acid. नहीं, अब राइबोसोम के ऊपर आपको अमीनो एसिड बैठता है, then it will be converted to peptide. I have written chain को polypeptide कहते हैं. नहीं, I have written stop codon, then I have written UTR, but nobody asked. नहीं, UTR. So in the mRNA, the structural genes which we are taking, in that this UTR is also present. What is this UTR? This UTR is nothing but untranslated region. The region which is not involved in protein synthesis. They doesn't want to code. There are few codons. They don't want to code for protein synthesis. No. For example, see, this is one UTR. For example, this is one UTR. Yes, did you understand? So to undergo with the translation process, protein synthesis करने के बाद से first thing we want translational unit we have taken. Number two, we want ribosome. Without ribosome, protein synthesis will not take place. Why? Because proteins are required. नहीं, energy होना ना. So proteins are required, and second, the R, that is ribosomal RNA is also required. Two things we have taken. Then one more very important part we want is third one, tRNA. tRNA, third part, tRNA is required. So here, what is this tRNA, sir? Yes, transfer RNA. Yes. So the basically the work of the tRNA is to take the information, transfer to the ribosome. That's all. So what information, sir? The work of the tRNA is to catch amino acid in the cytoplasm of a cell. There are so many amino acids present. In our cell, we have cytoplasm. In the cytoplasm, amino acids are present. What tRNA in short doing? It will catch the amino acid, it will give to ribosome. So that ribosome has to revise, study, know about the amino acid. Coding will be done. Okay. Finally, that amino acid will be converted to polypeptide or protein molecule. Is that clear? So here tRNA will do that work. Is that clear? So here when we study the structure of tRNA, basically when we study the structure of tRNA again it is linear. How mRNA is present, tRNA is also same. But when we study the structure, no, clover leaf model, in your books it is given clover leaf model, that is 2D structure. Is that clear? If you study 3D structure, inverted L shape. Okay. Why that shape is being formed, sir? Usually what happens, see for example, codon is present on the tRNA, UUG. Now, next to it, if the complementary codon, one more codon is present, AAC. Nay, that means UUG is complementary to, so it will go and bind. It will go and bind. Therefore, if you see, the structure is like this. Nay, it is binded like this. So that binding is done because of complementary base pairs present nearby. Is it clear? If you study in detail about the tRNA, cloverleaf model, no? So usually it has 5 arms. It has 5 arms. So this arm is called amino acid binding arm. Okay. We can tell acceptor arm also, in short, acceptor arm. So here the base will be present CCA. So once the amino acid comes, it will bind to this codon, CCA codon. Is that clear? Number one, first arm. Second arm, here. The second arm is called as ribosome binding site. This arm is called as anticodon. This is extra loop. Okay. And this arm is called as amino acid recognition site. Okay. But in your books, it is given as D loop. Yes. T loop. Correct? Yes. Why? Because in this dihydroxyuridine is present in excess. And here T intersection C. That is thymidine and anti-uridine is present in excess. Okay? Here a small loop is present where the 10 to 12 nucleotide bases are present. If there is any requirement, necessity, somewhere the codon, the bases are required, nucleotide bases are required, this extra arm will supply. Is that clear? And this arm is called as anticodon arm. What is that anticodon arm? In the mRNA, codons are present. In the mRNA, codons will be present. So anticodon will be present in the TR. Where this tRNA will go and sit, go and bind, where the codon is matching with the anticodon. Is that clear? So three things are required. Number one, translational unit that consists of mRNA, start codon, stop codon, peptide formation site, and UTR. Okay? Second, ribosome, eukaryotic ribosome. Then third, tRNA. Done? Shall we proceed with the process now? If you recall the transcription process, right? How the process started? How the process started? What was the first step called? Yes, initiation. Yes, initiation. Second step, elongation. Yes, third step, termination. Done? Yes. Same thing will happen in the translation also, but this initiation first step consists of number one. Number one, binding of amino acid, then charging of tRNA. Charging of tRNA. So what will happen, sir? So usually the amino acid is present. The amino acid is present. tRNA is also present. That amino acid has to go and sit on the arm. I showed you the first arm, amino acid acceptor arm. No, it has to go and bind. So here, binding will happen by its own. It will not happen. Some energy is required. No, some source of energy is required. So here, ATP will be involved. ATP will be involved in the presence of enzyme. In the presence of enzyme. Assume this is the enzyme. So first step will take place. Charging of amino acid. Charging of amino acid. The amino acid has to be activated first to go and bind with the tRNA. Yes. So for the binding, first we are charging the amino acid. How? By giving the ATP. So when this amino acid will take the ATP, no, it will not take full ATP. Full ATP. ATP में क्या है? Adenosine triphosphate है. नहीं. 3 phosphates are present. Yes or no? So here 2 phosphate will be released. 2 phosphate will be released and utilized by the amino acid. It will be utilized by amino acid in the presence of enzyme. Which enzyme? Amino acyl tRNA synthetase enzyme. Yes, amino acyl tRNA synthetase enzyme. What will be the product now? Yes, simple. We have taken amino acid. We have given energy in the presence of enzyme. Which enzyme? Yes. Nay. What will be the product? Amino acid is charged. Yes, amino acid is charged now. What is the leftover product? In 3 ATP, 2 phosphate molecules are released. What is remaining? Yes. What is that? AMP. Nay. What is that AMP standing for? Adenosine monophosphate. Yes, adenosine monophosphate plus enzyme complex. Done? Yes. First step, activation of amino acid, binding up of amino acid. Okay. Now, to whom it will go and bind? This will bind to tRNA. Second step. This will bind to tRNA. Nay. So when it binds to tRNA, then tRNA will be charged. That is your second step. Once the amino acid is charged, now it will go and bind to tRNA. Nay. So here, this amino acid which is charged now. Nay, it will go and bind with the, assume this is your tRNA. No? So this will go and bind with the tRNA. Yes, that is second step. Charging of tRNA. This AMP and enzyme complex, what it will do, sir? It will leave the process. Okay. When these two are binded together, now there is no work of AMP and enzyme complex. So they will leave the process. Okay. Now the step, two steps which are involved is the first phase of translation, that is initiation. Once the binding up of the amino acid to the tRNA and charging of the tRNA has taken place, that means the process will start now. Yes or no? The process will start. To start the process, what is required now? Yes, we want first, number one, we want mRNA. We have taken mRNA. Yes, whatever we discussed till now practically we are doing. Okay, we have taken mRNA. On this mRNA, what is present? UTR is present. Start codon is present. Stop codon is present. Yes. Poly A tail is present. Cap is present. Since we are discussing about eukaryotes. Okay. So I have drawn with the cap. I have drawn with the tail to the mRNA, and the structural genes are ready. Is it clear? So again, so here the different codons are present. That has to be coded. This is the region. This is the region that has to be translated now. Done? Any doubts still here? It is easy. You are able to understand? Yes, first thing we have taken. Second, what we have to take? What is required? Ribosome. Nay. So, shall I take the ribosome? This is the larger subunit. This will be smaller subunit. Done? Second. So, here, two RNAs are ready now. One is mRNA and one more is RNA. Ribosomal RNA is present in ribosome. Ready? Done? Now, third. Which one? Third one. tRNA. No? Now, the tRNA comes here. Yes. When it is coming, as I told you, in the ribosome we have three sites. Yes or no? Which are the three sites? 1, 2, 3. A site, P site and E site. Okay, EPA, EPA. Three sites are present. No? So here the tRNA, molecule, no, when it is coming in, first it will bind to the A site. It will bind to the A site. So that means the amino acid accepting site. Amino acid accepting site. So the amino acid is accepted here from the tRNA. The tRNA has transferred. What is the work of tRNA? Transferring work. Nay. So from where this tRNA amino acid was taken by tRNA? From the cytoplasm. From the cytoplasm it has taken. Nay. Now it is giving to ribosome. It is giving to ribosome. So here in the A site, the amino acid is present now. Is that clear? Now, the amino acid which is given to the A site, from the A site, this amino acid will go to P site. Okay. What is P site doing? The work of the P site is to convert amino acid into peptide. it has to convert amino acid into peptide polypeptide chain. One by one it has to convert. Is that clear? So here the AMP which has left there, nay, in the initiation process, the AMP have left the process, no? That means it is not involved. One phosphate is remaining here. Yes, one phosphate. So that phosphate will be utilized by the P site. It will be utilized by P site. Why? What it will do? The coding will be done between the amino acid and the amino acid on the tRNA is having anticodon. It is having anticodon. So here, when the tRNA is binding to this, no, if it is AUG here, it will be UAC here. Yes or no? So tRNA has binded. So that coding will give that this will be coded by the amino acid methionine. It will be coded by methionine. Coding is done. One amino acid got converted to the peptide. Is that clear? A protein molecule. So here, this amino acid is free now. Empty. The amino acid is empty. Why? Because this amino acid was transferred to P side. In the P side, amino acid will get converted to peptide. So first amino acid was coded as methionine. Is that clear? Now after coding, the tRNA which was present, it will come to the E site and it will exit. It will exit. समझ में आ रहा है? Yes, if you understand, very easy. What is happening in the whole process? ठीक है? mRNA is getting prepared. mRNA is ready. RNA is ready. tRNA is also ready. tRNA will catch the amino acid which is present in the cytoplasm. It will supply to the ribosomal RNA. Is that clear? And what this mRNA and rRNA is doing, it is converting the amino acid into polypeptide. That's all. Amino acid को protein में convert कर रहा है. That is the work done. So here, first start हुआ सुनिए. First AUG से ना start हुआ सो. That is initiation. It is initiation. So one tRNA has left. One tRNA is left. Now the ribosome will move to the next. The ribosome will move to the next codon. It is moving to the next codon. Again in the ribosome we have A site, P site and E site. Yes. Again one more RNA molecule will come with one more amino acid. Yes, it will be coded. For example, if here it is CCU, then there it will be, what it will be present? GGA. Yes, it will be coded. Assume the second, this amino acid will be sent to the P site. For example, phenyl alanine was coded. Yes, the RNA will come to the E site and it will come out of the process. Yes, so second polypeptide is formed. So to join this polypeptide, one by one polypeptide is getting formed. May that has to be joined to form a protein molecule. That joining work will be done by this monophosphate. Peptide bond has to be formed. That formation of bond between this polypeptide will be done by that monophosphate left earlier. Is that clear? So continuously if the process is going, no? Elongation. Yes, to start the process, sir, directly it has started. No, here factors are also required. In the eukaryotes, now some factors are also involved. Directly the process can take place, but few factors are involved. During the start, we have IF1, IF2, IF3. What is that IF1? Initiation factor 1, initiation factor 2, initiation factor 3. Okay. So same thing will continue. One RNA is coming, one tRNA is coming with one amino acid, binding with the ribosome, sending the amino acid to A site. From the A site, it will move to B site. P site is a site where the amino acid will get converted to polypeptide. Then after polypeptide is formed, tRNA will come outside of the ribosome. Okay. So assume it is elongating one by one, the codon is going on. Then we are reaching in the second phase, that is elongation. To move on this process, again we want factors. Which are that factors? EF1, EF2, EF3. Elongation factor 1, elongation factor 2, elongation factor 3. Okay. Now, sir, the ribosome is moving from one codon to another. What is the process called? One more question. The It is not elongation. Continuously, if it is moving from one codon to, we are telling elongation. Okay. That's a middle quagga process. Then it is elongation. If the ribosome is moving from one codon to another, again what happens? See, it will move to the next codon. It will move to the next codon. Yes or no? One by one it is coding. So, yes, initiation. No, initiation start hua. I am asking you, this ribosome, when it is moving from the one codon to another codon, what it is called? One more question. It is called translocation. It is locating from one place to another, no? It is called translocation. Remember, one more question. Translocation. Okay? Yes. Then you can see it is moving one by one, one by one, it is moving. So finally, wherever the stop codon is coming, whether it is a UAA or UAG or UGA. Okay. In these three, anyone is present. If it is present here, that means the process will come and stop here itself. To stop, again, we have three factors: RF1, RF2, RF3. Releasing factor 1, releasing factor 2, releasing factor 3 are involved. So the process will be stopped there itself. Is it clear? So until the process comes to an end, the chain of polypeptides will be growing. Okay. The chain of polypeptides is continued. Methionine, valine, serine, leucine, isoleucine, tryptophan. Continuously the polypeptides are present. Now, when all these are joined together, this will become one protein molecule. Done? Did you understand? Any doubts in this? समझ में आया? Yes, it's a bigger process, but if it is asked 5-mark question, it will be asked. No doubts? Yes? Everything is clear? Yes. I will ask you one question. I will ask you one question. Okay. We took mRNA, one mRNA we took. We took one ribosome, ne? And we took one tRNA. So one tRNA, after the work is over, it is coming outside. No? Whether only one ribosome is completing the whole process, or is there any process where multiple ribosomes come together to do this process? Yes? Only one is required. There is no any process where multiple ribosomes at a time they will code for many. Yes? Yes? Only one mono. No? Yes. It is possible that at a time many ribosomes also can code together. Okay. If only one ribosome is coding, mono. If many are coding together, we will call that ribosomes as polyribosomes. Polyribosomes. Remember, one more question. What is polyribosomes? Okay. When many ribosomes are coding in the translation process, they are polyribosomes. They are called polyribosome. Done. So here, now, finally, once the process is over, the termination process has taken place. Why? Stop codon has reached. Once the stop codon comes, the process has to stop. Is it clear? So once it is reached, what will happen? Yes, everything will be separated, released. For that release, we have three factors: releasing factor 1, releasing factor 2, releasing factor 3. Three factors are releasing it. Yes or no? Yes. So what will happen? The mRNA will be separated. Yes. mRNA will be separated. Your ribosome will be separated. Yes. Your tRNA will be separated. Yes. And finally, the polypeptide chain, no? That also will be separated. So ultimately, the end of the product after the process, what we are getting is protein synthesis. Protein synthesis. And in our body, no, in our body, hardly 2% of our genome is involved in protein synthesis. Not all. Complete cells are not involved. Only 2% of our genome are involved in protein synthesis. Is that clear? Did you understand? Yes. Any doubts you have in this process now? Yes. We know transcription process, very important. Translation. It's a continuation. Okay? In transcription, we are making RNA. In translation, we are making protein, not DNA. Protein. ठीक है, DNA replication में बनाते सो. If you want to make DNA from DNA, it is DNA replication. ठीक है. If you want to make RNA from the DNA, it is transcription. If you want to make protein from the DNA, it is translation. Done? Did you understand? Yes. So when we study translation, one very important topic we need to discuss is gene regulation, gene expression. Done? Regulation of gene expression. Any idea what is this regulation of gene expression? Yes. Now we have understood translation, no? Yes. To understand translation better, we need to study this topic. Regulation of gene expression. What do you understand by this word? Expression of genes? Nay, that means the genes are functioning something. Yes, the genes are functioning something. No, they are showing some expression. So that will be regulated. That means control. Control of gene expression. Yes, very important topic. Control of gene expression. To understand this, two scientists by name Jacob and Monod. Jacob and Monod. No, they have conducted, they have discovered, they have studied one concept called lac operon concept. Done? Lac operon concept. Again, this is also a 5-mark question. Very easy. If you understand, very easy question, 5 marks easily. They will ask you directly, they can ask you explain lac operon concept, or for 3 marks, they will ask you explain switch off mechanism or explain switch on mechanism. Half of the answer. Is that clear? Shall we proceed? Yes. Now, so here, as I told you, regulation means control. Controlling the gene expression. Now, example you take fan. The speed of the fan is controlled by regulator. Yes or no? Regulator. No? Similarly, when we talk about the lac operon, what is this operon? So here, cluster of genes. Cluster of many genes. Genes का bundle. ठीक है? Cluster of genes. बहुत सारे genes हैं. Cluster of genes. Now, that cluster of genes controlled by two things. Controlled by two things. One is regulator and second is operator. Two things. Ne? So, cluster of genes controlled by regulator and operator together makes operon. What is operon bole to? Cluster of genes controlled by, yes, the whole gene action is controlled by two things, regulator and operator. Did you understand till here? Now, this lac in this concept is lactose. ठीक है. अब generally, in general, what is this process, sir? What exactly is happening? So here, assume the bacteria is present. ठीक है. Why we are not telling about ourselves? The reason is eukaryotic lack of concept or any other concept is very complicated. ठीक है. So here, what Jacob and Mona did, he took E. coli bacteria and he has studied lacopron. So we are discussing about prokaryotes. ठीक है. So if you take a bacteria, E. coli bacteria, the E. coli bacteria is present in this environment. It is present in the environment. So for their survival, they want energy. That energy, how they will get? By taking glucose. Nahin. Assume, for example, I have kept a potato here. Directly potato here. That potato is the source of energy. कई को potato के अंदर glucose है. When the bacteria sits there, it will take up the glucose and it is surviving. It will continue to grow. So if the glucose is present, what will be the reaction? If glucose is absent, what has to be done? Some other molecule is present. If glucose is present directly from the potato, it will take. No, it is surviving. Now instead of in this environment, potato, I have kept a bowl of milk. Milk is present. So if the bacteria enters into the milk, it is getting glucose. Directly glucose है उसके अंदर नहीं है. Lactose है. नहीं. Lactose is present. So what bacteria has to do? It has to do transcription also. It has to do translation also. Then finally that lactose will be broken down into glucose and galactose. ठीक है. So that in two mediums, presence, absence. ठीक है. We will discuss after namaz. ठीक है. Till here I'll stop. After namaz we'll continue. to this concept. So now we know what is opera. We know what is opera. We know what is regulator. What is operator. Regulate, regulate the process. Control the process. We want to regulate it. And to operate, to run the process smoothly, operator is that one. That's clear. Now we will take one condition where the bacteria wanted to produce protein to get the superconductor. Okay. So two conditions in the medium where the bacteria is trying to make the protein molecule in the presence of lactose, in the absence of lactose. That is, assume milk is present here in our surrounding, in our environment. Now the bacteria is entering into the milk. Okay. When it is entering into the milk, it will take up lactose. Okay. If lactose is not present, then what will be the condition? Okay. Two conditions. So first we will take the DNA segment. We will take the DNA segment. So in this DNA segment, first we will go with the structural genes Z, Y. These three are structural genes. Okay. Now, so totally 7 blocks we have to make. Okay. Structural genes, that means cluster of genes, 3 different genes we have taken, okay, along with the operator and regulator. Along with the operator and regulator. Together, what it has become? It has formed operon. Okay. Structural genes with regulator and operator. Okay. Together, it is called operon. Together, it is called operon genes. Okay. Now, so here we have promoter. I have taken 2 promoters. Yes, I have taken a DNA where operon is present along with the promoter. Operon present along with promoter. What is present in the operon? In the operon, we have structural genes, we have regulator, we have operator. Okay. Yes. Yes. Done. Shall we proceed now? Yes. So now coming to the process, to go with the process. So here three structural genes are present. Three structural genes are present. These three structural genes consist of enzymes. It consists of enzymes. Which enzymes? It consists of enzyme beta galactosidase, permease, trans acetylase. Three enzymes produced by three structural genes. Done. Now, so here first medium, we are taking where lactose is absent. Lactose is absent. So assume the bacteria is present here. The bacteria is present in this environment where lactose is not present. That means there is no other growth source for the bacteria to survive here. Other molecules are also present. Nay? Other protein molecules are also present. If the bacteria is sitting on the desk, it can eat the desk. Yes. It is sitting on your body. It is trying to enter into your body. Yes. So some are the other source it will take. from some or the other source, it will try to grab the protein and it will try to survive. Is that clear? So first condition we are taking, lactose is absent. Done. Now the RNA polymerase, assume this is the RNA polymerase enzyme. No, this RNA polymerase enzyme will come and bind with the promoter to start the process. ठीक है. What process will take place? पहले mRNA बनना, उसके बाद tRNA, नहीं, उसके बाद rRNA. RNA formation होने के बाद it will go for the protein synthesis. So first transcription will take place, translation will take place, ultimately protein will be produced that bacteria will take. ठीक है. This is happening in the absence of lactose. Medium में lactose नहीं है, नहीं. So in the medium where lactose is absent, when the RNA polymerase enzyme comes and binds with the promoter is the region where the process starts. नहीं. So it will start with the process. So this is the regulator gene. This is the regulator gene, or we can also call it as inhibitory gene. Done? Yes. The promoter will tell the RNA polymerase to go and bind with the regulator gene to start the process. Okay? So the process will start. First, transcription will take place. So when the transcription is completed, what is formed? mRNA is formed. mRNA is formed. Is that clear? Now, after transcription, the mRNA will enter into second process, translation. Done? Translation is done. What will be the product form? Yes, protein. Protein. Done? यहाँ तक समझ में आया? Yes. Now, so who has done this translation and transcription process? Which gene? Yes, regulatory gene. I told you the first word regulator का तो controller. नहीं. So when we talk about regulatory gene, the regulatory genes are classified into two. Some genes will show positive effect, some will show negative effect. Okay? Positive control, negative control. So here, the gene which we have now, the regulatory gene which we have, which have made the mRNA and protein, it is showing negative control. It is having negative control. So here, the mRNA which is formed, we will call it as repressor. Repressor mRNA. Repressor का माना क्या? जो भी चीज को सप्रेस करता है. ठीक है? Anything which suppress is repressor. So the protein which is formed is which protein? Repressor protein. Yes. यहाँ तक समझ में आया? Yes. Now the repressor protein molecule will be taken by. It will be taken by. Continue करना ना process. It has to be taken again by the operator. नहीं. It should be taken by the operator, and operator has to continue the process. It has to move to Z gene, Y gene, A gene, and finally that protein molecule will be broken down into simple molecule, and that will be utilized by the bacteria. Okay? The molecule which we have, that has to be finally completed into glucose molecule. Okay? That will be taken by bacteria. But if the lactose is absent in this medium now, there is no lactose present in the medium where this process is happening. So here, the protein molecule which we have got is repressor protein. Now this repressor protein which is produced, it will go and bind with the operator. Okay? It has to go and bind with promoter. Why? To continue the process. Again, it has to take promoter, process will start, then it will move on, it will take operator, then it will go to structural gene, then finally protein molecule will be formed. But here what happened? The repressor will go and bind to the operator. Once it binds to the operator, it is blocking the operator. It is blocking the operator. So when the operator is blocked, further no transcription will take place, no translation will take place, no protein molecule will be formed. No polypeptides will be formed, no protein molecule will be formed. So process has stopped in the between itself. Nay, it is not finished. It is stopped in the center. Therefore, this process is called switch off mechanism. Switch off mechanism. Did you understand? Switch off mechanism. So what are the things that has happened? This has happened in the absence of lactose. Did you understand? Yes. Now, second medium we are taking where lactose is present. The medium where lactose is present. Yes. क्या हो रहा? खुश हो जा रही ना, उससे पूछ रहा हूँ. बोले तो सब जने 1-2 मिनट खुश हो जा सकते हैं. नहीं, कहती भी जोक बोल को रहे तो नहीं. ऑलरेडी बोरिंग क्लास से, बोरिंग टॉपिक नहीं. यस. नहीं सर, मेरे दांत अच्छे हैं, उसे दिखा रही हूँ. सो वो कहती भी है कि वो भी नहीं. फिर यस, लिसन. So second medium we are taking where lactose is present. So again I have taken the DNA segment where promoter, regulator, promoter, operator, 3 structural gene Z, Y, A. Yes. Now the function of Z gene is it will produce an enzyme called beta galactosidase. Y gene permease and A transacetylase. You have to remember these 3 names. Why? The main function is played by that 3 enzymes only. Okay. Now see, the RNA polymerase enzyme will come. RNA polymerase enzyme. Yes, it has binded with the promoter. The promoter will tell the regulator to start the process. Yes, the regulator will start the process. It will undergo transcription process. mRNA is produced. Which mRNA? Repressor mRNA. Then it will lead to the next process. Translation. Protein is produced. Which protein? Repressor protein. Yes. Now this protein, which is a repressor protein molecule, no, in the absence of lactose, what it has done? It has binded with the operator. No. But here, when the lactose is present in the medium, sir, how that lactose which was present in the medium was taken by this protein molecule? Here, these 3 enzymes will play the important role. Okay. The permease enzyme, permease है नहीं, permit करता है. Permease enzyme, it will permit the lactose to enter into bacteria. नहीं, I give you the example. Milk में lactose है. नहीं, bacteria entering into milk. अब वो lactose क्या करता है? Supply करता है. Lactose से bacteria के अंदर supply हो रहा है. Who will do that? Permease enzyme will do. ठीक है? So that lactose which was present in the medium will enter into the bacteria. It will enter into the DNA of the bacteria and it will reach here. It will reach here with the support of permease enzyme. Hoga? Did you understand? Now, this lactose, when it reaches the protein molecule, repressor protein molecule, this lactose will act as an inducer. It will act as inducer. Inducer का काम क्या? Repressor का repress कर रहा है. नहीं सप्रेस कर रहा है इंड्यूसर it will bind ठीक है it will bind and it will suppress the repressor इन्हें repressor को suppress करता है so here this inducer lactose molecule will come and it will bind to the repressor protein so therefore this protein is now not able to move to the operator to bind it is induced there itself nee? Now what happens the promoter is free operator is also free therefore the promoter will tell the operator to continue the process it will tell the continue the process again what happens the transcription will take place by and translation will take place by ZYA gene all the three structural genes will involve in the function and they will produce finally when we have taken lactose in the medium what will be produced lactose है तो और lactose ना बनता है नहीं so more amount of lactose is being produced so finally what is the product we got lactose yes we got lactose but bacteria doesn't want lactose no it is a complex molecule bacteria want glucose what has to be done so again what happens these two enzyme beta galactosidase enzyme and transacetylase enzyme which are present no they will break this lactose breaking down work okay they will break down the lactose into glucose and galactose and that will be taken up by the bacteria and they will survive is it clear so here since the binding of the inducer to the repressor protein took place the process did not switch off it was continued again it was on nay it was on from of the operator has operated the process and continued the process till the final translation has been done so this process is called switch on mechanism switch on mechanism whole concept is called lac operon concept okay that is lactose operon concept it is not only lactose any molecule you can take fructose you can take glucose sorry uh, maltose you can take tryptophan any of the process where the any particular medium is present and they are producing more amount of that is called as operon concept is it clear operon genes regulating themselves controlling themselves and they are supporting the process is it clear the whole process is gene regulation regulation of gene expression example is lac operon concept done directly they can ask you for 5 marks explain lac operon concept both you have to explain when lactose is absent the medium is stop switch off mechanism when lactose is present finally the glucose and galactose will be produced and it is switch on mechanism separately if they ask you 2 or 3 marks they will ask you explain the mechanism involved in switch off process in lac of or switch on in the lac operon concept done any doubts you have did you understand yes so today two very important topics we have discussed one is translation one more is lac operon concept done yes we'll solve some questions or we'll continue with the topic क्या करेंगे हाँ कंटिन्यू करो यस एक टॉपिक करा जेनेटिक कोड करा डाल तो हाँ लेटर विल गो अभी खाली उसके बाद दो टॉपिक बच को रहता है ठीक है यस so actually we need to study genetic code before translation ठीक है but why I am doing after translation when we know translation definitely we know what is genetic code also why because the codons which was carried by the tRNA was decoded by the mRNA codons okay and finally the amino acid were synthesized yes or no already we know something about codon now we know something about amino acid nee? Now when we study genetic code it will be easier what is this genetic code in one word anyone can tell me yes what is this genetic code yes no we can tell a form of language no that we are able to see in DNA and RNA. In DNA, we are able to see four bases. What are that? A, T, G, C. Yes or no? In RNA, we have A, U, G, C. Yes. So here, the language to study the codons. What makes a codon? Three nucleotide bases makes a codon. Okay. So genetic code is the language to study what are the codons that are present in DNA and RNA, and this codon to which amino acid it will code. To which amino acid? Decoding of amino acid by the codon present in the DNA and mRNA. Is that clear? This makes your genetic code. So this genetic code was first discovered. It was made by a person, a scientist by name George Gamow. George Gamow. Okay. So what he has done when he made the genetic code. Okay. He has taken these four bases which are present in the DNA and which are present in the RNA, and he tried to explain that there are 20 essential amino acids present in the organism body. Okay? There are many amino acids which are taken from outside, but essentially functional amino acid which are required by our body. Okay? So that 20 amino acids are present in our body, that will be coded by codon. Yes or no? So here 20 amino acids are present, coded by a codon. Codon has three bases. Nay. Yes. So what he did, he gave us a system called triplet codon system. Triplet. According to triplet codon system, all the 20 amino acid will be coded. It will be coded by the codon. Is that clear? By the codon. How many codons are present, sir? If you calculate three codons, nay, you have seen the structure of genetic code. Yes, we need to take your AUGC in three forms. In your book it is present. Nay, so we will take AUGC. Nay, first you should come UAGC. Yes. So similarly we will take here also AUGC. Nay, so again here in the third AUGC, AUGC. So like this, if we make 1, 2, 3, 1, 2, 3. If you tally, see, we are taking A. From here, we will take A. From here, A. Nay, again, I will take A. A. From here, I will take U. So, like this, the genetic code is framed. So, when the genetic code is framed, totally we will get 64 codons. We will get 64 codons. How? So, here 4 are present now. 4 into 4 into 4. The number will be 64. Nay? But that was not matching. 20 amino acids are there. When a codon codes, nay, a codon is having 3 bases. It will be 3 into 20. How much? 60. But here it is coming 64. No? So here experimentally how that 64 has occupied and what is the function of that was explained by two more scientists by name Nirenberg and Harbind Khurana. He is from India. He is from India. Done. So finally, when they conducted the experiment and when they made the code, so finally they told the the codon which is coding amino acid, no. So here 3 into 20, 60 codons will be framed. So 64, why one extra? So here one will be your AUG, which is playing a dual role. It is acting as a start codon also, and it is coding also. Okay, one. And in this whole genetic code, the language where the RNA and DNA is coded by the amino acid. No, so here three are stop codon or nonsense codon. Which are that? U A A, ne? U A G U G A, ne? So remove this. You can remember like this: Aga, A A G G A, Aga. Yes. Did you understand? Yes. Now, so when we study about this genetic code, we need to study the characteristic features or silent features. Very important. This will not be asked. Okay. In this topic, what is being asked? The question will be asked, write the salient features of the genetic code which is framed. No? So what is the salient feature of that genetic code? Or what are the characteristic features of the genetic code? So number one, the genetic code is triplet in nature. Point number one. Number two, the genetic code is universal. The genetic code is dual in function. Number four, the genetic code is commaless. Number five, the genetic code is non overlapping. Number six, the genetic code is non ambiguous. And number seven, the genetic code is degenerative. And number eight, the genetic code is having the start and stop codon, that is initiator codon and terminator codon. If they ask you for 3 marks, write the salient features. If you just write these salient features with one example, that is more than sufficient. Done? Shall we proceed now? Yes. Now, whatever the genetic code we make, complete 64 codons we have. In any of the codon, just now I showed you. नहीं, for example, यहाँ एक U है, यहाँ एक U है, यहाँ से भी एक U लिया हमें. So it will form U U U. नहीं. So like that continuation. If you take for example here it is a U C A G. That means again U U C. Then U U A U U G. Yes. So all the 64 codons if you take, is there any one codon with 4 bases, 5 bases, 2 bases? No. The whole genetic code is having triplet. No. Therefore we can tell triplet codon. Okay. Another sense here, if you take only 4 codons, right? If one amino acid is coding for the codon, it will be 4 amino acid. Right? If it is double, 4 into 4, that is 16. That is also not filling up the genetic code. If you take 4 into 4 into 4, 64. 20 amino acid if it is coding for all the codons, 61 plus 3, 64. Totally it is matching, no? So therefore, all the complete genetic code is having 64 codons. That means each and every codon is having 3 nucleotide bases. Done? Genetic code is triplet in nature. Number one. Second, genetic code is universal. It is universal. Here, few points. 99% they are correct. 0.1% it is wrong. 99.9% correct. 0.1% it is wrong. So that if you take the ratio, 99 is more. 0.1% or 1% is nothing. Right? If you take 99.1% or 99.2%, 99, what you will tell? You will tell 100%, sir. Yes or no? So here, for telling, for explaining the universal, genetic code is universal in nature. No, whatever the codons that we have, they are universal in nature. This is applicable for the first one. If I tell AUG, nay, AUG. So any organism you take, any organism, you take plants, you take animals, you take bacteria, you take humans, nay, in any DNA, any organism DNA, the start codon is AUG. Yes or no? And here this AUG will be coded by methionine. It will be coded by methionine. Is that clear? It will be coded by methionine. So now any bacteria, any animal you take, no, whether it is your cell, whether it is my cell, AUG is coding for methionine. Okay. If you take bacteria, prokaryotes, there it will, this will be coded by formyl, again methionine. So therefore, F met methionine or methionine both are same. Nay, but 0.1% what I told. Nay, it is correct. Universally, it is correct now. But if you take protozoans, if you take protozoal DNA, or if you take mitochondria of any cell, okay, bacterial cell or protozoal cell or fungal cell, okay, in the mitochondrial context, the DNA segment which is present there, AUG is not getting coded as the start codon, or it is not coded by methionine. Okay, in the mitochondrial matrix, as I told you, in the mitochondrial matrix, and in the, what I told, yes, protozoans, किसमें आती हैं प्रोटोजोन्स? किसमें आती हैं? कौन से किंगडम में आते हैं? सो it will come under second kingdom, protista. Nay, yes or no? Chrysophyte, dinoflagellates, slime molds, euglenoids, and protozoans. So in the protozoans and the mitochondrial matrix, the AUG is not coding for methionine. Is that clear? Then what it will code, sir? AUG is not coding only. In them, in the protozoan and in the mitochondrial matrix, AUG is not a start codon. Instead of AUG, GUG is a start codon. And this GUG is coding tryptophan. It is coding tryptophan. Okay? But this is exceptional. हमें उसे नहीं लेते, छोड़ डालती. ठीक है? 99% similarity is there. Therefore, we will tell genetic code is universal for all the organisms. Is that clear? Yes. Then genetic code is showing, some codons are showing dual function. Nay, again, if you take the example of AUG here, what AUG is doing? Number one, it is acting as a start codon, initiator codon, and simultaneously it is coding for methionine. Is that clear? Yes. Then genetic code is commaless. Whatever the segment you take, whether it is a big segment or small segment, the code will be continuous. Nay, U C A G C C U. So here there is no comma. There is no comma in between. Therefore, genetic code is commaless. Okay. And genetic code is non-overlapping, means assume see U C A A G I have written. That means it is a complementary base. This will not come in the genetic code, in the DNA segment. This will not happen. If it is UUC present here, here it will be CCA. There will be no overlapping. If it is complementary, क्या होता है? अगर इन्हें इसका complementary रहा तो it will bind, overlap. ऊपर आपको लग जाता ना. So there will be no overlapping also. Therefore, genetic code is non-overlapping. Then genetic code is non-ambiguous. What do you understand by this word non-ambiguous? You heard this meaning, you know the meaning of ambiguous? No? In the dictionary, if you see, the meaning of ambiguous is not clear. Ambiguous बोले तो not clear. It is not clear. अब I am telling genetic code is non-ambiguous का तो genetic code is clear. Genetic code is Very clear. Completely the 64 codons which are present, each and every codon is explained. It is non-overlapping, it is comma-less, it is universal. Yes? Ne? And what we need to understand here, these two things, non-ambiguous and degenerative. Ne? To understand this here, ne? one codon will code one amino acid. At a time, one codon will code only one amino acid. एक ही बार एक कोडॉन ही कोड कर सकता सो नहीं. अब हमें ट्रांसलेशन प्रोसेस करे कितने कोडॉन्स कोड करे एक बार को 21 ना पॉलीपेप्टाइड बना सो एक अमीनो एसिड बाइंड करे सो नहीं. सो one codon will code for one amino acid. That means it is clear, non ambiguous, clear है. ठीक है. It is degenerative. Genetic code is degenerative, means इसका opposite. इसका opposite क्या? See here, one amino acid will be coded by many codons. It will be coded by many codon. For example, first U है ना, U ये दो, ठीक है. Two codons coding phenyl alanine. They are coding phenyl alanine. Is that clear? So here, one amino acid coded by many codon. That means genetic code is degenerative. Is that clear? Sensible gati. It will code for particular amino acid. A particular codon. Ab देखो UUC, UUU, two different codons, but they will code for phenylalanine only. Whether it is a prokaryotic, whether it is a eukaryotic bacteria or fungi or plant cell or animal cell, it is coding for phenylalanine only. So it is degenerative. Did you understand? Yes. Last point. Genetic code is having initiator codon and terminator codon. Initiator codon is a start codon. कौन सा? AUG. And terminator codon 3. UAA, UAG, UGA. They are called as terminator codon or nonsense or stop codon. Why? Because they are not doing any functioning. They are stopping the process. Is that clear? When this codon will come, whether it is transcription process going on or translation process going on, it has to stop. Therefore, it is called stop codon. Done? Did you understand? Any doubts you have? Yes. So three topics we have discussed: translation, Lacan concept, genetic code. Done? Two topics are remaining. One is human genome project and DNA fingerprinting. Done? We'll continue in the next class. Take down few questions. As the column 1 with column 2. Match the column 1 with column 2. Column 1, A, termination. A, termination. B, translation. C, transcription. D, DNA replication. Then column 2, number 1, amino-acyl tRNA synthetase. Amino-acyl tRNA synthetase. Number 2, Okazaki fragments. Okazaki fragments. Number 3, GTP dependent release factor. Number 4, RNA polymerase. RNA polymerase. Option 1, number 1, A2, B3, C1, D4. A2, B3, C1, D4. Option 2, A1, B4, C2, D3. Option 3, A3, B1, C4, D2. Option 4, A2, B4, C1, D3. What will be the answer? The match. Option C. Termination. GTP dependent release factor. Translation. Amino-acid tRNA synthetase. Transcription. RNA polymerase. DNA replication. Okazaki fragments. The repressor protein. The repressor protein binds to the operator. Binds to the operator of lacopron. Of lacopron. Number 1, lactose is unable to remove. Lactose is unable to remove the repressor. Number 2, RNA polymerase is activated. Number 3, structural genes Z, Y, and A fail to transcribe. Number 4, galactosidase does not act on the lactose. does not act on lactose. Yes, answer D. Galactosidase does not act on the lactose. Arbind Hargobind Khurana Hargobind Khurana was awarded Nobel Prize for number one, discovering of transposons. Discovering of transposons. Number two, artificial gene synthesis. Artificial genes synthesis. Number three, dye-separating. Dye-separating of genetic code. Dye-separating of genetic code. Number four, nucleotide sequence of tRNA was discovered. Nucleotide sequence of tRNA was discovered. What will be the correct answer? It is option C. Dye-separating of genetic. What do you mean by dye-separating of genetic code? Yes, breaking. Yes. What do you understand by the word dye-separating? Hmm? The codons were made. Yes. In the genetic code, if you see, the codons are coded by particular amino acid name. So which codon will code for which amino acid? The 20 amino acid are fitted in the genetic code. That means decoding of the amino acid by the particular codon was given by Arvind Khurana and Hiranbach. Therefore, the Nobel Prize was given to them. Because dye-separating means decoding. The smallest part of the DNA, the smallest part of the DNA that undergoes recombination, that undergoes number one, muton. Number two, cistron. Number three, replicon. Number four, recon. is recon. Mutant, that is involved in mutation process. Cistron, they are functional genes. Replicon is involved in replication, and recon is recombination. In a DNA, the percentage of thymine, the percentage of thymine is 20. Is 20. What is the percentage of guanine? What is the percentage of guanine? Number one, 20%. Number two, 40%. Number three, 30%. Number four, 60%. Yes, in DNA, the percentage of thymine is 20%. What is the percentage of guanine? Yes, A, T, G, C. A and T डबल बॉन्ड जी एंड सी ट्रिपल बॉन्ड नहीं तो अब ए एंड टी में 20 है 20 20 40 का तो जी एंड सी में कितना रहता है 30 30 60 इक्वल टू 100% तो गुआनिन में कितना रहता है 30% रहता है match the following genes match the following genes of lactoferron match the following genes of lactoferron with their respective products with their respective products. Option A, column 1. Option A, hygiene. Hygiene, that is regulatory. B, Z gene. C, A gene. D, Y gene. Column 2. Number 1, beta galactosidase. Number 1, beta galactosidase. Number 2, permease. Number 3, repressor. Number 4, trans-acetylase. Match and tell me. I gene, I gene, repressor. Z gene, C gene, Y gene, trans-acetylase. ZYA. Z is for, Y is for, Y is for permease, and A is for trans-acetylase. Few more questions we'll solve in the next class. Okay, so after finishing the chapter, some more questions we will solve.